Actually, today I will present our recent findings entitled Targeting Sulc Family Kinase, a Promising Strategy for Inhibiting Cancer Metastasis. In this presentation, I will share how radiotherapy, although now is widely being used for the treatment of solid cancer, can promote, paradoxically, the cancer metastasis, especially reshaping of the tumor microenvironment. And also I would like to show the potential role of Sulc family kinase in this type of responses and the pharmacological value of each inhibitors. Radiotherapy is one of the common cancer treatment options, and it can be applied to about 50% of solid tumor patients at least once during their therapy. Actually, the high energy ionizing radiation directly can make DNA strand breaks and also indirectly interact with the water molecule to generate reactive oxygen species. This generation of the ROS can also cause the DNA and cellular component damage. So radiotherapy has been continuously developed over the years. Actually, the introduction of the 3D conformal and intensity modulatory radiotherapy can make precise tumor targeting. However, the radiotherapy impact on normal tissues cannot be completely avoidable. Actually, direct exposure to the normal tissue not only limit the therapeutic doses of radiation, but also lead to the chronic tissue damage, such as fibrosis. In case of lung tissues, the exposure of radiotherapy to pulmonary epithelial cell initially trigger injury response through immune cell activity change. Firstly, infiltration of monocyte to alveolar sac and differentiation into the macrophages, and then fibroblast activation and eventually accumulation of the extracellular matrix near the alveolar sac. All these events can lead to pulmonary fibrosis and even irreversible lung injury. Several reports, such as one nice paper in Nature Cancer 2022, demonstrate that radiation-induced normal tissue damage can make a tumor-friendly environment in the lung. This radiation-induced environment increases the risk of recurrence and metastasis by increasing metastatic colonization in non-cancerous tissue. So, while radiotherapy may provide short-term therapeutic benefits, in the long term, it can also contribute to the development of refractory lung cancers. However, the exact mechanisms remain unclear, and therapeutic targets to inhibit this process are still limited. Based on this background, we hypothesized the following point. First, radiotherapy can make a tumor-friendly microenvironment through fibrosis. Second, radiotherapy-induced fibrosis is mediated by monocyte-derived macrophage infiltration and subsequent M2 polarization. Finally, we want to find out the exact target of this process. So, we suggest Sulc family kinase is an important mediator of radiotherapy-induced fibrosis, and then we want to show whether SFK-targeted agent prevent radiotherapy-induced pro-tumorigenic microenvironment formation. To test this hypothesis, we first established the clinically relevant radiotherapy mimicking mouse model system. For the lung radiation model mouse, the thorax was exposed to X-ray radiation. For SBRT, stereotactic body radiotherapy mimicking model, the beam-guided radiation was only focused on 0.2 centimeter scale region of the thorax. Finally, for liver radiation model, the abdominal cavity of the mice was exposed to X-ray radiation. So because of the immune cell activity differences, we utilized two different mouse species, BL6 and valve-C. And we also used two different mouse cancer cell lines, B16F10 melanoma cells and 41 mammary tumor cells. Both the cell lines stably overexpress luciferase, and so we can nicely detect metastasis of cancer cell to the viable animal models. Actually, the 2 times exposure of the X-ray radiation could not change survival rate and body weight gain. But surprisingly, we found that metastasis into the lung, so in other words, the engraftment of cancer cell to lung tissues was highly increased by radiation exposure here. And also, it can aggravate survival of the mice. All these data indicate that initial radiotherapy exposure can create pro-tumorigenic, the niche formation. Then, how did radiation cause this type of responses? Micro-CT analysis showed no change of the lung volume after radiation exposure. However, the histological analysis revealed that the fibrosis, as evidenced by the alveolar collapse and Masson's trichrome staining positive areas increase, was significantly enhanced by radiation. Moreover, the mRNA expression levels of the several kinds of the pro-fibrogenic genes, such as TIMP1 and alpha-smooth-musclectin and COL1A1, their all mRNA levels was significantly enhanced by radiation. So we can conclude that radiation exposure induces early-stage lung fibrosis. Then we performed public data set analysis of lung cancer patient exposed to radiotherapy, shown in left figure. Actually, several fibrotic marker genes were upregulated in irradiated the human lung cancer tissues. And their expression was really positively correlated with poor patient survival. So overall, radiotherapy induces early stage lung fibrosis, which can make tumorigenic the niche formation. Then we would like to know the underlying mechanism for this phenomena. So we performed flow cytometry analysis, and then we found that among the many kinds of the immune cell type, only the macrophage population was significantly enhanced by radiation exposure. And again, the enhanced macrophage population was characterized by the CD206 positive M2 macrophage phenotype. Actually, lung tissues contain two major macrophage populations. First one is tissue-resident alveolar macrophage, and second, monocyte-derived macrophages. So we examined their respective roles in radiation-induced pulmonary fibrosis. By using the serial isolation protocol, we found that radiation enhanced the infiltration of monocyte and monocyte-derived macrophage. But tissue-resident macrophage population was not changed by radiation exposure. Again, M2 polarization was selectively enhanced in monocyte-derived macrophages, but not in tissue-resident macrophage population. We addressed the next question. Which kind of mediator is important for this type of responses? So we performed RNA sequencing analysis and found that in irradiated lung tissues, the several cytokine and chemokines, their mRNA expression was highly unregulated. Particularly, the TGF-beta, unknown the key mediator for fibrosis, is obviously unregulated during the early phase of the radiation injury, just on day 2. Again, ELIJA confirmed the real secretion of TGF-beta-1 the 2 days after radiation exposure. The next question, which type of cell type is important for the radiation-induced tumor-friendly environment formation? Surprisingly, we found that the direct exposure of the radiation to bone marrow-derived macrophage itself enhanced the M2 macrophage population, as evidenced by CD206 positive population increase. Again, IgA1, a representative the M2 macrophage phenotype marker, their expression level was highly enhanced by radiation exposure here. And finally, we also found that mRNA and protein secretion of the TGF-beta1 was also nicely enhanced by X-ray irradiation, direct exposure of the BMDMs to X-ray irradiation. So, finally, we would like to know, the causal. We further examine the causal role of the macrophage population in the radiation-induced tumor-friendly microenvironment formation. For this purpose, we performed reversal experiment by using 2 different technology. First one is bisphosphonate treatment. Actually, zoledronate non-selectively depleted macrophage population. Again, we also used F480 neutralizing antibody to inactivate macrophage. By using these 2 different methodology, we found that the lung metastasis of the melanoma cells was significantly inhibited by zoledronate treatment and again F480 neutralizing antibody treatment. And also, the mouse survival rate was significantly improved by these 2 different approaches. However, T-cell influenced PD-1 therapy could not change all these type of responses. So as the interim summary, actually the infiltration of and M2 polarization of the monocyte-derived macrophage, which is accompanied by increased secretion of TGF-beta1, underlie the mechanism of radiation-induced early-stage fibrosis. Then we changed our focus to the Sulc family kinase. By using the human transcriptome and protein atlas data cells, we found that all kinds of the Sulc family kinase expression was enhanced by the radiation exposure in human cases. And actually, Sulc1 and YES1 were ubiquitously expressed in all type of cells in lung tissues. So we compared the correlation of the Sulc and YES1 mRNA expression and pro-fibrogenic, the marker gene expression. We found that positive correlation between Sulc1 and YES1 expression and pro-fibrogenic gene expression. And here, right here, okay. In previous slide, I already showed TGF-beta1 was highly unregulated the 2 days after radiation exposure. Just the same as this one. We found that mRNA expression of the Sulc and YES1 was nicely unregulated by the radiation on day 2. Moreover, the SMAD2, the downstream target of the TGF-beta1 receptor, its phosphorylation as well as Sulc family kinase phosphorylation was enhanced in irradiated lung tissues. So we concluded that Sulc family kinase could be one of the contributors to radiation-induced fibrosis. Actually, Sulc family kinase is a group of non-receptor tyrosine non-receptor tyrosine kinase regulating diverse cellular process. Actually, as shown in left figure, Salk family kinase transduce signals from the upstream oncogenic receptors to downstream targets. And so it can be considered as a proto-oncogene. Furthermore, the dasatinib developed by BMS has been approved for the myeloid leukemia. So, in human lung epithelial cell line, in Bears-2B cells, the TGF-beta stimulation caused Salk family kinase phosphorylation at 3-hour time point, which promote upregulation of the fibroblast activation marker gene expression and again EMT marker gene expression. And when we knock down Salk or YES1 by siRNAs, we found that the fibroblast activation marker gene expression and again the EMT marker gene expression was significantly inhibited by the YES1 and Salk1 knockdown. So then we further used pharmacological inhibitor of Salk family kinase. As inhibitor of the kinase activity, we used dasatinib. For the inhibitor of the scaffolding and kinase activity, we used brand new NXP900 compound developed by Nuvectis. Although I didn't show them a full data set, actually the TGF-beta1 induced Salk family kinase activation was nicely suppressed by NXP compound. And again, the fibroblast activation marker gene expression and EMT marker gene expression was also significantly abolished by the NXP900 compound. Again, one of the typical phenotype of the fibroblast activation, the cell migration, TGF-beta1 induced cell migration was also completely inhibited by dasatinib or the NXP900 compound treatment. So all these data indicate that pharmacological targeting of SFK effectively inhibited fibroblast activation. Then what about the in vivo effect of SFK inhibitors? In our model system, we found that radiation-induced lung metastasis was almost completely inhibited by NXP900 administration. And again, also improved mouse survival rate. We further used other mouse species, Barb-C, we got same result. Even when you use T-cell and immune-deficient nude mouse system, we can get same result. All these results showed that SFK targeted agent efficiently prevent the formation of the radiation-induced the pro-tumorigenic microenvironment formation. Furthermore, the Marsan trichrome staining, which represent the collagen selective staining method, also showed that the radiation-induced lung fibrosis was completely reversed by NXP900. However, M2 polarization, radiation-induced M2 polarization was not changed by NXP and SFK inhibitor. Again, the macrophage infiltration was rather enhanced by SFK inhibitor. This data suggests that potential target point of the SFK should be fibroblast and epithelial cells, myofibroblast or fibroblast activation. And also this compound actually did not act on macrophage infiltration and TGF-beta release from the macrophage. So all this type of result imply that SFK targeted therapy should be a useful strategy to prevent radiation-induced pulmonary fibrosis and tumor-friendly environment formation. Actually, the radiotherapy also can be applied to the abdominal abdominal malignancy such as liver cancer and colorectal cancer beyond lung cancers. So what about radiation impact on other organs and the pharmacological effect of Salk family kinase inhibitors? For this purpose, we used the radiation model, abdominal cavity radiation exposure model system, shown in left figure. Actually, radiotherapy to abdominal cavity induced mild hepatic injury. Again, the liver fibrosis was marginally enhanced by X-ray irradiation. And also we got the same result found in the lung tissues. Also, macrophage population was significantly enhanced by X-ray irradiation. Then, in the spleen liver metastasis model, we found that X-ray irradiation to abdominal cavity really increased, the liver fibrosis, but just marginally, but highly enhanced, metastatic nodule formation in the liver tissues. And all kinds of these phenomena can be reversed by NXP900 SFK inhibitor administration. So this data show that radiotherapy promotes liver metastasis through a pro-tumorigenic microenvironment formation. And during this pathway, SFK play a key role to mediate the lung and liver fibrosis. Then finally, we expanded our work to the bone metastasis of breast cancer. Above 70% of patients with metastatic breast cancer actually develop bone metastasis. To mimic this condition, we used intra-arterial injection model system. Actually, just 1 week after, we found that the 41 cells is mainly distributed in the bone tissues. And surprisingly, we found that NXP900 effectively reduced breast cancer metastasis without affecting the primary tumor growth. So SFKs also serve as a critical target in preventing bone metastasis of breast cancer cells. Because of time limitation, I could not show them all data set, but briefly, interaction of the Salk with focal adhesion kinase is important for the regulation of the actin remodeling. And this process can cause metastatic cancer cell motility. And second, Salk is also involved in the osteoclast differentiation. And this process can make directionality of the cancer cells to bone tissues. So final conclusion. Actually, we discovered a new pharmacological target, Salk family kinase. In the formation of the radiotherapy induced the tumor-friendly microenvironment formation. Furthermore, we confirmed that the SFK targeted agent should be effective options to prevent the formation of the pro-tumorigenic microenvironment formation. All these findings highlight a new combinational approach integrating SFK inhibitors with radiotherapy to improve long-term outcomes in cancer patients. Actually, first radiotherapy, the part work was performed by my previous PhD course student, Dr. Yongjun Choi. Now he's working at Harvard University as a postdoc. And the rest part now maybe we will submit this paper in just 1 month later, 2 months later, was performed by my master course student, Minju Kwon. Thank you for your attention.